Mendel's laws are the foundation of classical genetics. The whole chapter is about how traits pass from parents to offspring in a predictable way and why one gene or two genes can create such clear patterns. The visual frame makes that purpose obvious. Heredity is not random chaos, it follows rules. That is exactly why Mendel matters so much in biology.
The real question behind genetics is simple. Why does a mango seed grow into a mango tree? And why do offspring resemble parents but still show differences? Humans noticed variation long before the science of inheritance was understood. They used natural variation for artificial selection too, such as breeding wild cattle into the well-known Sahiwal cows. So the problem was familiar even before the explanation was available. Mendel gave that explanation. Three words form the vocabulary of heredity: genetics, inheritance, and variation. Genetics is the field of biology that studies these processes. Inheritance is the process by which traits are passed from parent to progeny. Variation is the degree to which progeny differ from their parents. That distinction is important because many students mix these terms together, but they are not the same thing. Genetics is the subject, inheritance is the transfer, and variation is the difference. Mendel was the mastermind behind classical genetics. He worked in the mid-19th century, and his experiments on garden peas ran for 7 years, from 1856 to 1863. That long duration is one reason his conclusions were so strong. He did not guess the rules of heredity. He built them through repeated observation and controlled breeding. The slide's warning about dates is worth remembering exactly, because exam questions love to change the numbers slightly. Mendel's method was revolutionary because it combined mathematics and biology. He used large sample sizes and followed successive generations of test plants. That gave his conclusions scientific credibility. He was not just observing one or two plants. He was analyzing patterns across many offspring. The key insight here is that heredity became a measurable science through Mendel's approach. That is why his work still sits at the center of genetics.
Mendel chose the perfect specimen, garden peas. Why peas? Because they show clear, contrasting traits with no confusing middle state. You can easily see tall versus dwarf, yellow versus green, smooth versus wrinkled. That clarity made statistical counting possible. The stronger the contrast, the easier it is to identify dominance, recessiveness, and ratios. So the plant itself was part of the genius of the experiment.
The foundation of Mendel's work was true breeding lines. An NCERT true breeding line is one that, after continuous self-pollination, shows stable trait inheritance and expression for several generations. Mendel selected 14 true breeding pea plant varieties, which together represented 7 pairs of contrasting traits. That count is a classic trap. There are 14 varieties, but only 7 contrasting character pairs. The self-pollination diagram shows how these traits remain stable through generations, which is exactly what made the parental lines reliable. The first 4 contrasting traits involve seeds and pods. Seed shape can be smooth or wrinkled. Seed color can be yellow or green. Pod shape can be inflated or constricted. Pod color can be green or yellow. The color flip is a classic examiner trap. For seeds, yellow is dominant. For pods, green is dominant. So dominance does not follow one universal color rule across all characters. You have to learn each pair separately. The next 3 traits involve flowers and stem. Flower color is violet or white. Flower position is axial or terminal. Stem height is tall or dwarf. These are clean binary traits, and that is exactly why Mendel could build solid ratios from them. The traits are sharply opposed, which makes the inheritance pattern visible without ambiguity. In Mendel's experiments, that clarity was everything.
The experiment begins with a P generation cross. Mendel took two true breeding plants with contrasting traits, such as tall and dwarf, and carried out hybridization after removing the anthers and collecting pollen. The table of Mendel's 7 contrasting traits on the right is the key anchor: stem height, flower color, flower position, pod color, seed shape, and seed color. The big warning is simple: there is no blending here. A pod is either inflated or constricted, never somewhere in between.
The monohybrid cross shows how a single character behaves across generations. In the P generation, true breeding tall is crossed with true breeding dwarf. The F1 generation is uniform and all plants resemble the dominant parent, so the dwarf trait seems to disappear. But after self-pollination, the F2 generation brings the hidden trait back in a 3:1 ratio. That is the first major Mendelian message. The recessive factor is not destroyed, only masked. Mendel's factors are what we now call genes. Each gene exists in alternate forms called alleles. In the diagram, one allele codes for the dominant trait, like tallness, and the other codes for the recessive trait, like dwarfness. The pro tip on the page is worth keeping exactly in mind. Use capital letters for dominant alleles and lowercase letters for recessive alleles. That simple notation makes the whole chapter much easier to track. The genetic vocabulary becomes very precise here. Genotype is the genetic blueprint, while phenotype is the physical expression. Homozygous means identical allelic pair, like capital TT or small tt. Heterozygous means dissimilar allelic pair, like capital T, small t. These 4 terms are the language of heredity. Once they are clear, the rest of Mendel's laws become much easier to understand. The law of dominance explains what happens in a heterozygote like capital T, small t. One factor dominates the other. So the dominant factor is expressed in the F1 generation and the recessive factor remains hidden. The important insight is that the F1 plant looks physically identical to the dominant parent, even though its genotype is different. That difference between appearance and genetic composition is one of the central lessons of genetics. The law of segregation is shown through the meiosis funnel. In a diploid parent with genotype capital T, small t, the 2 alleles separate during gamete formation. Each gamete receives only one allele, so a heterozygote produces 50% capital T and 50% small t gametes. The page also gives the probability logic. Segregation is random, and every pollen grain or egg has an equal chance of carrying either allele. That 50-50 split is the foundation of all monohybrid ratios. The Punnett square is the analytical tool Mendel's logic relies on. It is a graphical method for predicting the probability of all possible genotypes in a cross. The layout is simple. Write one parent's gametes across the top and the other parent's gametes down the side, and then fill the inner squares. The tool was introduced by British geneticist Reginald C. Punnett, and it remains one of the easiest ways to map inheritance outcomes. The Punnett square for the F2 generation gives the classic monohybrid result. The cross capital T small t times capital T small t yields capital TT, capital T small t, capital T small t, and small tt. That produces a genotypic ratio of 1 to 2 to 1 and a phenotypic ratio of 3 to 1. The observation on the page is very important. TT and TT are physically indistinguishable from the outside. That is why phenotype alone cannot reveal genotype in dominant traits. Hello. The famous 1 to 2 to 1 ratio in genetics isn't just biology. It's actually mathematics in action. Each gamete has a 1/2 chance of carrying capital T and a 1/2 chance of carrying small t. So when they combine, the cross behaves like a binomial expansion. Open bracket, 1/2 capital T plus 1/2 small t close bracket squared. When you expand this expression, you get 1 capital TT, 2 capital T small t, and 1 small tt. That hidden mathematical pattern is what gives Mendel's ratios their remarkable precision. In the end, genetics here is really just probability applied to biology. The genetic blind spot is a classic problem. If a plant shows a dominant phenotype, how do you know if it is capital VV or capital V small v? Just looking at the outside is not enough. A tall violet plant could be homozygous dominant or heterozygous, and the phenotype will not tell you which one it is. That is the point where phenotype stops being enough, and another test is needed. The solution is the test cross. A dominant unknown is crossed with a known recessive parent instead of self-crossing. The recessive parent acts like a genetic scanner because it contributes only recessive alleles, so the hidden genotype of the unknown parent is forced to reveal itself in the offspring. That is why the recessive parent is so useful in diagnosis. The test cross outcomes are very diagnostic. If the unknown is capital VV, then crossing it with small vv gives 100% capital V small v offspring, all showing the dominant trait. If the unknown is capital V small v, then crossing it with small vv gives a 1-to-1 ratio of dominant to recessive offspring. The rule of thumb is clean. A 1-to-1 ratio in a test cross proves the unknown parent is heterozygous.
The final summary ties the one gene story together. The monohybrid phenotypic ratio is 3.1, the genotypic ratio is 1.2.1, and the test cross ratio is 1.1 for a heterozygote. The law of dominance says one factor masks the other in a dissimilar pair, and the law of segregation says alleles separate randomly during meiosis. That is the blueprint of one gene inheritance, and it is the base on which the rest of Mendelian genetics is built. Now, we begin with the classic Mendelian idea of dominant and recessive factors, then move into the law of dominance, the law of segregation, and then into the real-world glitches that make inheritance more interesting. Incomplete dominance, codominance, multiple alleles, and the idea that some traits are shaped by more than one gene. That gives the full roadmap for monohybrid, dihybrid, and non-Mendelian inheritance.
The first baseline is the law of dominance. Mendel called them factors. Today we call them genes or alleles. These factors occur in pairs, and in a dissimilar pair, one factor can dominate the other. That is why in the F1 generation, only one parental character is usually expressed, while the hidden one shows up again in the F2 generation in the familiar 3-to-1 ratio. The key idea is simple. The dominant trait masks the recessive trait in a heterozygous condition. The mechanism behind dominance is easier to understand if you think of the gene as an enzyme factory. A normal, unmodified allele produces a working enzyme, and that enzyme produces the dominant phenotype. A modified allele may produce no working enzyme at all, so the recessive trait appears only when both copies are broken. That is why the phenotype often depends entirely on the functioning of the unmodified allele. If the factory works, the dominant trait is seen. If it fails, the recessive trait comes through. The law of segregation is the purity protocol. Alleles do not blend into one another. They stay separate and are recovered as such in the F2 generation, even if one of them was hidden in F1. A heterozygous parent produces two kinds of gametes in equal proportion, one carrying the dominant allele and the other carrying the recessive allele. That 50-50 split is the heart of segregation. This is the reason Mendelian inheritance stays predictable instead of becoming a mixing bowl.
Incomplete dominance is the first major glitch. Here, one allele is not completely dominant over the other, so the F1 generation shows an intermediate phenotype. The classic example is red and white snapdragon flowers giving pink flowers in F1. In the F2 generation, the phenotype ratio becomes 1 red, 2 pink, 1 white, and that matches the genotype ratio exactly. That 1.2.1 pattern is the signature of incomplete dominance.
Codominance works differently from incomplete dominance. Here, both alleles express themselves completely in the same individual. In the ABO blood group system, when the alleles IA and IB are present together, both remain active. That is why the red blood cell shows two different surface sugars at the same time. The blue markers represent the sugar produced by allele IA, while the orange markers represent the sugar produced by allele IB. Neither allele suppresses the other, and neither gets blended. Both traits appear together side by side. So the blood group becomes AB, where both parental characteristics are expressed clearly in the same individual. The ABO blood group system is a classic example of multiple alleles. Here, three alleles control one character, IA, IB, and I. When a person has IAIA or IAI, the blood group becomes A because allele IA is dominant over I. Similarly, IBIB or IBI gives blood group B since IB also dominates over I. When IA and IB come together, both express equally, producing blood group AB. This is codominance, where both traits appear side by side. Finally, blood group O appears only when both alleles are I, written as II, because the I allele does not produce any surface sugar. A simple way to remember this is to think of I as a blank canvas. By itself, it shows nothing. But when IA or IB is present, their trait becomes visible. Here we encounter a very important perspective shift in genetics. Dominance is not a fixed or permanent property of a gene itself. It actually depends on which phenotype or visible character we choose to study. In this example, gene capital B controls starch synthesis in peas. First, let us look at seed shape. Plants with capital B and plants with capital B small b both produce round seeds. Only small b produces wrinkled seeds. So for seed shape, capital B behaves as completely dominant over small b. Now let us look at a different trait, grain size. Here, capital B produces large grains. Small b produces small grains. But capital B produces intermediate sized grains. That means the same gene now shows incomplete dominance for this particular trait. This is the key lesson. A single gene can appear to behave differently depending on the character we examine. Dominance is not an absolute property of the gene. It depends entirely on the phenotype being observed. The dominance spectrum brings all three patterns into one frame. In complete dominance, F1 resembles one parent and the F2 ratio is 3 to 1. In incomplete dominance, F1 is in between the two parents and the F2 ratio is 1 to 2 to 1. In codominance, F1 resembles both parents and the F2 phenotype ratio matches the genotype ratio. Again, 1 to 2 to 1. The NEET exam shortcut is this. If the F2 phenotype ratio is equal to the genotype ratio, think incomplete dominance or codominance. The examples here are pea plant height, snapdragon flower color, and human ABO blood groups. The inheritance of two genes shows how Mendel's rules scale up from one trait to multiple traits. In this cross, a plant with capital RRYY is crossed with a plant carrying small rRYY. The first plant is round and yellow. The second plant is wrinkled and green. During fertilization, the gametes combine, producing offspring with the genotype capital R, small r, capital Y, small y. These F1 hybrids are round and yellow because capital R dominates small r and capital Y dominates small y. The key idea is that this behaves exactly like two separate monohybrid crosses happening together. One gene controls seed shape, while the other controls seed color. At this stage, the two traits do not interfere with one another. They are simply combined into a single hybrid organism.
The dihybrid cross makes the mathematics of inheritance even clearer. When the F1 hybrid, capital R small r, capital Y small y, is self-hybridized, each trait separates independently during inheritance. Trait 1, seed color, behaves exactly like a normal monohybrid cross. 3/4 of the offspring are yellow, while 1/4 are green. At the same time, trait 2, seed shape, also follows a monohybrid pattern. 3/4 of the offspring are round, while 1/4 are wrinkled. The important idea is that these two traits are operating independently at the exact same time. The dihybrid system is really just two separate 3 to 1 monohybrid crosses running together. This principle is called independent segregation or independent assortment. That independence is what makes the combined dihybrid outcomes possible. Once these smaller pieces become clear, Mendel's inheritance stops feeling like memorized ratios and starts feeling like a logical structure you can reason through. The dihybrid cross makes the mathematics of inheritance even clearer. When the F1 hybrid, capital R small r, capital Y small y, is self-hybridized, each trait separates independently during inheritance.